Hello friends, welcome to Biology Extrema, where we teach biology from the basics to the extreme. Myself Keshav Bansal, and today we are going to study immunology. What we are going to study in immunology? We are going to study toll-like receptors. Okay, so let's take an overview that what this topic is all about. Okay, so we have this toll-like receptor. Okay, so first thing we can notice that it is a receptor. Okay, and what we are studying immune system. We are studying immunology, which concerned with immune system. So what happens that this receptor is it acts as a microbial sensor. Okay, it is a microbial sensor. Okay, what happens whenever there is a receptor? Okay, whenever there is a receptor, there is something we can call it a ligand, okay, which is going to bind to that receptor. Okay, in the case of immune system, we call this ligand as antigen. Okay, so, and we have it, this is our, for example, TLR, toe-lag receptor. Okay, or we can say microbial sensor, a type of microbial sensor. So what happens when it this is a receptor and it binds to an antigen. Okay, so what happens that this antigen, where this antigen comes from? Okay, this antigen comes from the microorganism. Okay, this antigen comes from the microorganism, or I can say pathogens. Okay, I can say pathogens, but specifically. Okay, so what happens that we have so many cells in our immune system. Okay, and these cells are constantly monitoring. Okay, these cells are constantly monitoring the body and looking for pathogens. Okay, these cells are constantly monitoring the body, thus roaming the body, and they are searching for the pathogens. Okay, and when they find these pathogens, okay, they find these pathogens in the form of antigens. Okay, we will give, in short, we will give a specific term to these antigens. Okay, so they search the environment. Okay, they constantly monitor it, and they monitor for what? They monitor it for the antigen, and that antigen is going to come from pathogen. And when they find these antigens, when they find these components of pathogens, okay, what they do, they produce a response. They produce a response. In what form? They produce this response in the form of alert. Okay, they alert the whole body. Okay, they alert the whole body. Okay, so they are acting kind of like sensors. Clear? And when they alert the body, what they generate? They generate an immune response. They generate an immune response. Okay, you can take an analogy of like a policeman who is looking with a torch. Okay, and searching for a thief, and whenever a thief is found, he kind of alert other policemen. Okay, so it's the same case that these toll-like receptors are type of receptors, okay, which are microbial sensor. They sense the environment for danger. What kind of danger? They search the environment for antigens, which comes from the pathogens. Okay, and when they find these antigens, they bind to it. Okay, and they create a response, which is an alert state, which generates the immune response. Okay, and now further action can be taken against this pathogen. Okay, so now we are going to give specific names to these, the names which are used to denote these antigens. Okay, we call them pathogen associated molecular patterns. Okay, I'm writing it, pathogen associated molecular patterns, or in short form we can refer to them as PAMPs. Okay, because what happens that some special type of molecules are only present in pathogen. See how we evolved and how pathogens evolved. Okay, and how our immune system evolved in the way that only some kind of molecules are only available on pathogens. Okay, they aren't found in the human body. Okay, so they are usually present in the human body. So what happens when these microbial sensors, okay, they search for the environment, and when they find these PAMPs, they know that some pathogen has breached the security, okay, has breached the defenses and enter the body. Now it's time to alert the body and start for the action, means triggering the immune response. Okay, so these are pathogen associated molecular patterns, okay, PAMPs. And these receptors, these toll-like receptors which acts like a microbial sensor, okay, we call them PRRs. We call them PRRs. Okay, what do we mean by PRRs? Pathogen recognition receptors. Okay, pathogen recognition receptors. So these pathogen recognition receptors senses the environment for what? For the PAMPs, pathogen associated molecular patterns. Okay, and these are not the only type of PRRs. There are many type of PRRs. Okay, like CLRs, like NLRs, like regulator receptors, MDA. Okay, so there are many classes of PRRs. Okay, today we are focusing on toll-like receptors. Okay, so toll-like receptors are class of pattern recognition receptors, PRRs. Now we understand this term. Okay, so what happens, how we discovered them? Let's talk about it. So it was discovered as a protein, because we know that receptors are what? Receptors are protein, and it is a integral transmembrane protein. Okay, so it is a protein which involved in the establishment of dorsoventral polarity in the developing Drosophila melanogaster embryo. What did it mean? That scientists were studying development of Drosophila melanogaster. Okay, and they found in the development that some kind of protein are responsible for establishing that some part. Okay, this is the body, this part is going to be the dorsal part, and this part is going to be the ventral part. Okay, so some proteins are responsible for this polarity, that this is dorsal part and this is ventral part. Okay, so the protein which are establishing this polarity, we call these proteins as toll-like receptors. Okay, and they are found in they were first discovered in Drosophila melanogaster. Okay, and also that in adult fly, these proteins, these toll-like receptors, like us, they are involved in resistance against fungal infections. Okay, like us, it is associated with immune system. Okay, so also in adult fly, it is associated with a defense mechanism. Okay, resistance against fungal infections. Now we are going to talk about its biochemistry. Okay, what kind of we know that it is a type of protein. Receptors are type of protein. They are type 1 integral transmembrane glycoproteins. Okay, we know that membrane proteins can be integral and peripheral. Okay, so it is a type of integral protein, and in the integral, it is a type of transmembrane protein. Okay, and not just a protein, it is a glycoprotein, means it has a carbohydrate moiety attached to it. Clear? So we know that in a transmembrane protein, we have an extracellular component. Okay, we have a membrane spanning region. Okay, we have a membrane spanning region, and also we have a cytosolic region. Okay, so what happens, that we know that that proteins are made of amino acids, and that's why a protein is going to have an N-terminal and a C-terminal. Okay, so what happens, that N-terminal is extracellular. Okay, this protein is consisting of amino acids. Okay, so N-terminal lies in the extracellular domain. Okay, while the C-terminal lies in intracellular domain. Okay, this is important, and this TLR is made up of 700 to 1100 amino acids. Clear? Now this N-terminal, if we talk about this N-terminal contains leucine-rich repeats. Okay, this L-terminal contains leucine-rich repeats in the extracellular region. Okay, which is made up of 24 to 29 amino acids. Okay, and these leucine-rich repeats are responsible for recognizing PAMPs. Okay, so recognition function is due to these LRRs, leucine-rich repeats. Okay, and they are found in the N-terminal, means the extracellular domain. Getting me? And if we talk about the C terminal, now this C terminal, okay, what happens when ligand is going to bind the LRRs? It is going to transmit the signal through the membrane spanning region, and then the membrane spanning region is going to transmit the signal through to the cytoplasmic region. Okay, the cytoplasmic domain, or you can say the intracellular domain, or we can say the C terminal domain, because we know in the case of TLRs, the intracellular domain, the cytosolic domain is the C terminal domain. Okay, so here we have TIR. Okay, we have TI, this is also TIR domain because it consists of toll interleukin 1 receptor. Okay, basically it further initiates biochemical signaling called TLR signaling transduction pathway. TLR signaling transduction pathway. So it kind of activate other components of TIR pathway, TLR signaling pathway. Okay, so and if you talk about the humans have 10 TLRs. Okay, 10 members, while in case of mice we have 12 members. Okay, in case of mice 10th TLR is absent. Okay, so 1 to 9, and then 11, 12, 13. Okay, so total 12. Claro. Now what happens that these TLRs, that they can be present on the cell surface, okay, on the plasma membrane, embedded in the plasma membrane, or they can be intracellular, means they are present on the membrane or some vesicle, okay, some organelle, like for example endosome. Clear? Why they are present such way? Because some pathogens are going to be extracellular. Okay, so for that cell surface TLRs are capable to detecting them, but some pathogens can be intracellular, like viruses. Okay, so to detect them, some TLRs are evolved to be intracellular. Okay, so in humans, which TLRs or cell surface TLRs 1, 2, 4, 5, 6 and 10, while TLR 3, 7, 8 and 9 are intracellular TLRs. Okay, and all these TLRs recognize different set of ligands which comes from different microorganisms. Okay, and here we have a chart. Okay, so you can even skip this if you don't want to go that deep. Clear? So like I was saying that that this TLR 7 and TLR 8, they recognize single stranded RNA, they recognize virus. Like TLR 3 recognize double stranded RNA viruses. Okay, so these are intracellular and they act on the ligands of viruses. If you understand the video, please like the video, subscribe our channel, share the video. If you can donate a small amount, it will help us in providing this noble service of providing free lectures and quality notes. So thank you from Biology Extrema. See you next time. Take care.